All right. Well, thanks for inviting me. So, like she said, my name is Donnie Musgrove. I'm a statistician at Medtronic. Little background: I've been a statistician in research and development at Medtronic for 5 years. We focus in my group on implantable cardiac devices. Um, and I realized as I've sat through this for a day and a half that there's not a lot about devices, and so our regulatory is our processes are quite a bit different than pharma, right? You got phase 1, 2, 3. We don't actually have that in our devices. So hopefully this can translate and you can find some use from this. So just a little background on the package. So I'm going to be talking about the package, the development process, and its ultimate use in a clinical trial. So I'm going to focus just a little bit on the actual utility of the package. So the bayesDP package, DP standing for discount prior, has several functions for combining historical or external sources of data into analyses common to clinical trials. And so the idea is that the weight given to external data is related to the similarity between the external and current data under consideration. So for example, top right, this shows up. Okay, top right, you can see this is a treatment group. We have some data we've collected as part of a trial, and it follows this kind of normal distribution. That's the green dashed line. Sorry, I hope this is colorblind friendly. And so it's got a mean of 45 here. And meanwhile, perhaps we had some data from a pilot study, or we have some historical data or some external data that we want to use in the trial. And of course, we have approval to do that. And so this data has a mean of 50. And so somehow we want to use this data. And so as we can see, a mean of 45 more and a mean of 50, we don't see a lot of overlap in these densities. So perhaps we don't want to use a lot of that data. And so in fact, the posterior falls right on top of the current data. So it's like we're not even using this historical data. And so the way this works in practice is we have what we call a stochastic comparison. So this is a comparison between the current and historical data. Takes values between 0 and 1. 1 is very similar. 0 is fairly dissimilar. And everything in between, it's kind of similar. And so in this particular case, this example, I've seen about 10%. So we've got a value of 0.1. And so this translates to a weight of 0.1 under this particular assumption. And we call this a discount function. This is how we take that stochastic comparison and translate it into a weight that we're going to apply to the historical data. So functions in this package include your usual outcomes. So binary, we can handle binary outcomes, regression modeling, linear regression via BDPLM, continuous outcomes similar to a t-test, and then survival modeling. And this is basically through the piecewise Cox model. Of course, we're in a Bayesian context, so that's how we usually do Bayesian survival. And I've got an example here. So BDPLM, right? It takes in a formula, and then you just point it to the data that you have. Okay, so getting to the package development. So this package, and actually this methodology began as a collaboration between industry and the FDA via a public-private partnership called the Medical Device Innovation Consortium. And so MDIC, this is basically they have a partnership between government and the medical device industry. And they're mainly focused on promoting innovation efforts, not so much on trying to get new policies. And so the basic concept here is Bayesian methods allow for including external sources of data and early stopping of trials via theoretically principled approaches. So you can imagine, I'm not exactly sure what sample sizes you're looking at in pharma, but I typically see sample sizes smaller than 400. So 400 is like the max I usually see. So we can see a lot of payoff if we can incorporate say data from a pilot study into our trial. So that could reduce the sample size. And then early stopping, I think everyone appreciates that. If I can stop the trial early, then I can get the device or the product to market faster. And so on the technical side, though, from the industry perspective, is Bayesian methods are difficult to implement. Even if you're a big company, I think Medtronic's fairly large, we still have a lot of pushback from clinical statisticians that they just do not want to implement these methods. They're hard to use, and typically you have to use an outside consulting company or take specialized software. So it's just a lot of work. And then at the same time, FDA perspective is that there's an increased burden to check these submissions. And so they have tight turnarounds. The submissions need to be well organized, and they need code that they can actually rerun and understand. So that was the goal with this bayesDP package, and it was to create functions that are simple to understand and leading to fast implementation. So now this was kind of, I would say, swimming along the development, the methodology, and the software. And it kind of came to a head when internally at Medtronic, a group decided to use this methodology for their renal denervation procedure. And so briefly, I'm just going to talk about renal denervation. So in this picture here, so this is a kidney, and a catheter is inserted through the urethra into the bladder and into the right here, the kidney, near the kidney. And it's kind of like an ablation if you're not familiar with it, but it derives, the tool delivers a radio frequency pulse to sort of kind of burn the tissue. And in this case, it'd be the renal nerve. And the goal is to reduce blood pressure in patients that have high blood pressure. And so unlike most device trials, usually we implant a device and that device stays in the body, right? So pacemaker, et cetera. In this particular case, this is a procedure. So it's an outpatient procedure. And so the trial itself to seek approval, it consisted of actually two single-blinded, randomized and sham-controlled trials. So one was called off-med. So these are patients that are not on medications but have high blood pressure, and then on-med, which is for patients on blood pressure medication. So at the same time as the procedure, or right before the procedure, the patients receive a renal angiogram, and to them, it feels a lot like receiving renal denervation. They can't tell the difference. Only the surgeon is actually unblinded, right? They do the angiogram and then they do the procedure. But the surgeon is not involved in follow-up. So both off-med and on-med trials utilize the Bayesian design. They both incorporate historical data from pilot studies, about 80 patients in each study, and they have pre-specified interim analyses. And so the final sample sizes really depend on basically how much the current data is related to the pilot study data, and also when enrollment stops, or when we can prove non-inferiority. So back to development. So early on in development, the decision was made to publish the bayesDP package to CRAN, and then any analysis could be linked to a particular version of the package. So I've got a screenshot here. This is the archive, starting way back in 2017. And of course, it doesn't show the current version, which was submitted earlier this year, but it's at version 1.3.4. So prior to submission, we needed some validation efforts. So we hired a external company to validate parts of the package that were going to be used in the trial itself. And this was BDPLM. So we had a covariate adjustment, adjusting for baseline blood pressure. And so on top of this, we needed speed. So we actually ended up recoding the entire function in C++ via RCPP Armadillo. Of course, if you're familiar, RCPP is for C++, but Armadillo is a linear algebra library. And so I wasn't heavily involved in the validation process. Of course, the external company was, but I do know we had back and forth with unit testing and integration testing to get things straightened out. But in the end, we felt it was successfully validated, and then it was submitted to CRAN. So kind of to wrap this up, the discount prior methodology, so the methodology itself and the implementation in our package was deemed acceptable by the FDA. It's ultimately used in both off-med and on-med. So off-med actually finished after the first interim analysis. Again, I think, 331 patients total, and they proved superiority. On-med, that remains ongoing, but I guess it remains to be seen how that's going to turn out. Have some time. So I just want to thank several people that helped guide the bayesDP package development. So Sean and Tarek, a lot of the early legwork for the code and the development of the methodology itself. Martin and Graham at Medtronic really pushed to use this method, or were able to use this method in the renal denervation trial. And then Ted Lustig, he was a big part of interacting with the regulatory. He's since moved on from Medtronic. And now if I have time for questions, please reach out to me via email. I'll be in the Slack chat for at least an hour or so, and then otherwise we can do a little Q&A right now.
All right, thanks, Danny, for your presentation, and great idea, by the way. If someone is pushing back on a method, just wrap it up in a package, validate it and serve it.
Yeah, exactly.
I end up there. We have one question and we'll have 1 minute. So how many historical studies can bayesDP handle at a time?
One. It's pretty much hard-coded to handle only one. So it's going to give you the weight for exactly one trial, and that's it. I mean, obviously it could be generalized. It's just not something that we required, and a lot of this was pushed by requirements.
Thank you so much.